Virus in question today is West Nile virus. Since according to virus classification, where viruses are classified based on phenotypic characteristics such as morphology, nucleic acid type, the mode of replication, host organisms, and the type of disease they cause, West Nile virus would be assigned to group 4, and it has an unassigned order and is part of the Flaviridae family, Flavirus genus, and West Nile virus species. West Nile virus is also part of arboviruses, which are diseases transmitted through arthropods, including flies, sandflies, lice, fleas, ticks, and mites. West Nile virus emerged as a distinct virus around 1,000 years ago and had two distinct lineages, 1 and 2. The U.S. strain is related to lineage 1. The genus Flavivirus also contains other species like Zika, yellow fever, dengue, and the tick-borne encephalitis virus, Palm Creek virus, Parramatta River virus. In humans, the central nervous system is affected, and West Nile virus can also infect mammal species and has also been identified in reptilian species, including alligators and crocodiles, and also in amphibians. The incubation period of the West Nile virus, that is the amount of time from infection to symptom onset, is typically between 2 to 15 days. Flaviruses, which the West Nile virus is classified under, replicates in mosquitoes, mammals, and birds during their natural transmission cycles. The genetic material of the West Nile virus is a single strand of RNA, which is between 11,000 and 12,000 nucleotides long. It is positive sense, meaning that the sequence is translatable into protein. In the replication process, the West Nile virus's envelope fuses with the cellular membrane of the host cell. The nucleocapsid, which is like the bag that contains the RNA, is uncoated and the RNA is released into the cytoplasm. Then the viral genome serves as a messenger and also translates the viral proteins as a template for the RNA replication. Then copies of the West Nile virus are packaged within new virus particles and are transported in vesicles, which are like tiny air sacs, to the cell membrane. The primary method that West Nile virus is transmitted is through the female mosquitoes, since only female mosquitoes feed on blood. In the U.S., a species of mosquito such as the Culex pipiens, Culex tarsalis, and Culex cinquefasciatus are the main vector species. Side note, vector species are any agent that carries and transmits an infectious pathogen into another living organism. Mosquitoes are infected with the West Nile virus by feeding on birds with the West Nile virus, and mosquitoes may also transmit the virus through vertical transmission, which is where disease-causing agents are spread from mother to offspring. In these cases, it may be through vertical transmission that mosquitoes are able to carry the West Nile virus through the seasons, such as winter. When the species that are infected with West Nile virus don't develop a sufficient viral level, they don't transmit the disease to uninfected mosquitoes, and so they are not considered major factors in West Nile virus transmission. What are some symptoms of someone infected with West Nile virus? Well, most people infected with West Nile virus actually don't have any signs or symptoms, and it's only around 20% of people who are infected that develop a mild infection called West Nile fever. And the common signs of symptoms of West Nile fever, they include fever, headache, body aches, vomiting, diarrhea, fatigue, and skin rash. If the West Nile virus progresses to be more serious, which happens in less than 1% of infected patients, the virus causes a serious neurological infection, including inflammation of the brain, which is called encephalitis, and of the membrane surrounding the brain and spinal cord, and that is called meningitis. Symptoms of that include high fever, severe headache, stiff neck, disorientation or confusion, stupor or coma, tremors or muscle jerking, seizures, partial paralysis, or muscle weakness. Although the signs and symptoms of West Nile virus usually last a few days, signs and symptoms of encephalitis or meningitis can linger for a few weeks or months. And the terrifying thing is that certain neurological effects, such as muscle weakness, can be permanent. In these severe cases, treatment consists of supportive care that often involves hospitalization, intravenous fluids, respiratory support, and prevention of secondary infections. Is there currently a vaccine for the virus or ways in which to prevent the infection? There is no vaccine, but the best way to prevent West Nile virus is to beware of mosquitoes. One should wear mosquito repellent with DEET, thiazotulamide, and one should also take precautions such as applying sunscreen first and then repellent, and being aware that mosquitoes that often transmit disease are most active during twilight periods such as the dawn or the dusk or in the evenings. Fun fact, 2 mosquito traps, one in Kitchener and one in Cambridge, have tested positive for the West Nile virus. The Region of Waterloo Public Health and Emergency Services report. The traps were set August 8th and 9th, and Public Health found out about the results on Monday, August 14th. A study that was published in 2016 titled Interleukin 17A promotes CD8 plus T-cell cytotoxicity to facilitate West Nile virus clearance, which shows that interleukin 17A promotes CD8 T-cell mediated clearance of West Nile virus, which had protected the mice in the study from lethal West Nile virus infection. In fact, in previous studies, CD8 T cells, which are also known as cytotoxic T cells, have been shown to already prove immunity against intracellular pathogens. And this particular study really focused on interleukin 17A and how it works together with CD plus T cells to regulate a diverse immune function to microbial infections, malignancies, and autoimmune diseases. This is also a novel function of the interleukin 17A in promoting CD8 plus T cell cytotoxicity. Cytotoxicity is defined as the ability of certain chemicals or mediator cells to destroy living cells. Another study was called Identification of Genes Critical for Resistance to Infection by West Nile Virus using RNA Sequence Analysis. This study was published in 2013. RNA sequencing is actually a very novel way of revealing the presence of RNA in a biological sample at a given moment in time. This study involved 10 healthy donors, and they were infected in vitro with West Nile virus. And from those samples, a total of 28 million reads were gained, as well as 1,514 transcripts. From that data, novel gene changes were detected, and some were found to be unique. These previously unknown genes that are associated with a West Nile virus resistance can hopefully play a critical role in controlling the viral infection. An immunostimulator called AMD 3100 has promise against West Nile encephalitis.